Organic Molecules by Bolon and Austin Deng. So what are organic molecules? Well, on this slide, organic molecules are molecules that consist of carbon atoms. Since carbon has 4 valence electrons, or electrons that can form chemical bonds with other atoms, carbon is often able to form 4 covalent bonds with other atoms, including nitrogen, oxygen, and more.
The binding of carbon atoms to these various molecules creates variety in molecular structure. Eventually, many carbon molecules with these other molecules are strung together to form macromolecules, complex molecules or polymers, or molecules that have a single unit of monomers repeated numerous times. Polymers are often formed through condensation, or the removal of water, and are broken apart through hydrolysis, the addition of water. The 4 most important macromolecules are carbohydrates, proteins, nucleic acids, and lipids, as shown to the diagram on the right.
Carbohydrates. One example of an organic molecule is a carbohydrate. Carbohydrates are formed by the linkage of chemically similar sugar monomers, also known as monosaccharides, to form polysaccharides. Their main role is to provide energy and are the body's main source of fuel. Everything from brain function, as shown in the bottom left picture here, and operations of the organs to waste elimination, as shown through the digestive system here, requires the work of carbohydrates. We will talk about structure of carbohydrates in a later slide.
Types of carbohydrates include monosaccharides, which are the monomers of carbohydrates and are pentoses and hexoses. Though they are monomers, both pentoses and hexoses are important to life. Hexoses all have the formula of C6H12O6 and include glucose, fructose, and other important molecules. Meanwhile, pentoses such as ribose and deoxyribose form the backbones of nucleic acids. Moreover, there are also disaccharides and oligosaccharides. Oligosaccharides are several monosaccharides bound together, and disaccharides are 2 monosaccharides bound together. Finally, we have large polysaccharides, which are large polymers of monosaccharides. These consist of starches, glycogen, cellulose, and more.
Disaccharides, oligosaccharides, and polysaccharides are all constructed from condensation reactions that form glycosidic linkages, or the removal of water that links monosaccharides together. Moreover, a ring-like structure is found in the monosaccharides of pentose and hexose, each 5 carbon and 6 carbon ring sugars. More importantly, polysaccharides have several important structures. The parallel structure is found in cellulose and makes plant cell walls extremely strong. Also, the branched glycogen and starch structures, as shown in the last 2 images below, are not as strong as cellulose and can be more easily broken down.
Proteins are the most varied and common molecules in living organisms and are formed from various combinations of many amino acids, the monomers of polypeptide polymers. Each of these amino acids are chemically similar to each other. The amino acids found in proteins all contain 2 functional groups, the amino group, the carboxyl group, and an R group. Lastly, they can be separated into 2 groups, structural and functional.
Protein types and functions. Protein can be functional or structural. Functional groups include enzymes that speed up biochemical reactions, defensive proteins such as antibodies that respond to invasive particles, hormonal and regulatory proteins like insulin that control physiological processes, receptor proteins that receive and respond to molecular signals within an organism, and storage proteins that store amino acids. Furthermore, structural proteins provide stability and allow for movement. These include glycoproteins, actin filaments, and connective tissues inside our bodies. Here are photos of both structural and functional proteins.
Protein structures. Proteins have a very complex structure and are formed through dehydration synthesis and amino acids that are linked together by peptide bonds. Overall, there are 4 levels of protein structure. The first is primary structure, which is the linear sequence of amino acids joined together by peptide bonds. The second is secondary structure, which are alpha helices holding amino acids in helical form and beta pleated sheets allowing the polypeptide chain to fold back and forth. The third is tertiary structure, which is the 3D shape of a protein twisted and folded due to different bonds between R groups, such as ionic, covalent, hydrogen, and etc. The last is quaternary structure, which is more than one polypeptide chain group.
Lipids. The next type of macromolecule we will be learning are lipids. Although they are often referred to as fats and oils, lipids are hydrocarbons, meaning molecules that are composed of carbon and hydrogen atoms. They are insoluble in water because of their multiple nonpolar covalent bonds, making them hydrophilic and staying together instead of mixing with water if two substances are combined. They are held together through van der Waals interaction forces. As a result, huge macromolecular aggregations can be formed where the lipid molecules are still distinct from one another, but strongly held together.
Lipid types. The most common unit of lipids is known as triglycerides, or simple lipids. Triglycerides, solid at room temperature, are known as fats. Those that are liquid at room temperature are known as oils. Triglycerides, like fats and oils, are also excellent when used to store chemical energy. Another type of lipid is known as the phospholipid. Phospholipids are similar to triglycerides in terms of structure. However, one major difference is that phospholipids have a phosphate component to them that creates a hydrophilic head on the phospholipid. As a result, they are amphipathic because of their hydrophobic tails, allowing them to create a bilayer in aqueous environments, where they are lined up in a sheet. Two molecules stick, with the heads facing outward towards the water, and the tails facing inward so that no water can reach the center. This is known as the phospholipid.
Lipid structure. Lipid structure is comprised of mostly carbon and hydrogen atoms, which is a reason why lipids are considered hydrocarbons. A triglyceride lipid is comprised of 3 fatty acid molecules and 1 glycerol molecule. Glycerol is a small molecule that contains 3 hydroxyl groups. Fatty acids contain long non-molecular hydrocarbon chains attached to a single polar carboxyl group. The long hydrocarbon chain on fatty acids is very hydrophobic as a result of the multitude of CH and CC bonds. A triglyceride has 3 fatty acid molecules attached to a glycerol molecule that is created through condensation reactions. Fatty acids on the triglycerides may also differ. Saturated fatty acids only have a single bond between the carbon atoms and the hydrocarbon chain. In unsaturated fatty acids, there exists double bonds in the hydrocarbon chain. Kinks in the hydrocarbon chain are caused by double bonds. These kinks can affect the properties of fatty acids.
Nucleic acids. Nucleic acids are molecules that are important to the reproduction and growth of cells and determine cell traits and the coding of proteins within cells. They are made of monomers called nucleotides. There are also 2 types of nucleic acids that each have different functions, DNA and RNA. DNA makes up genes to code for the production of proteins, which in turn causes cell activities. Meanwhile, RNA works with DNA in protein synthesis.
Nucleic acid types. The 2 types of nucleic acids are DNA and RNA. Both are formed through dehydration synthesis, or the cleaving of hydrogen from the phosphate of one nucleotide and a hydroxyl from the sugar of another, removing water and forming a covalent bond. DNA is made of the sugar deoxyribose and codes for protein production, while RNA is made of the sugar ribose and aids DNA and protein synthesis. There are 4 types of DNA nucleotides: adenine, guanine, thymine, and cytosine. An additional type, uracil, is found in only RNA. Also, adenine and guanine are longer, having a double carbon ring, and are known as purines, while cytosine, thymine, and uracil are shorter, having a single carbon ring, known as pyrimidine.
Nucleic acid structure. The difference between the structures of DNA and RNA are that DNA consists of 2 strands, while RNA consists of only a single strand. DNA consists of a double helix that is twisted around and is helical in shape, while RNA is linear. Both DNA and RNA have a sugar phosphate backbone that sticks out. Additionally, the complementary base pairing rule holds for both DNA and RNA, and adenine must pair to thymine or uracil in RNA, while guanine must pair to cytosine. This is because the width between the 2 strands of DNA molecules must be constant.